Cool. So I'm Brian. Thanks for coming, everybody. Hopefully this will be a good intro to the seminar series. But as Peter was mentioning, this has been the basis of my PhD over the past 4 years. It's kind of wrapped up now. So my project is entitled Enhancing Anti-Cancer Immunotherapy by Disruption of the Non-Receptor Tyrosine Kinase FES. And this meeting is being recorded. Cool. So just a brief outline of what we'll go over, some background, my hypotheses, results, future directions, et cetera, et cetera. And so starting off with some background, most people know about cancer. A lot of people in this room probably study cancer, but cancer is a really big disease in our population. It's the leading cause of death in Canada. Approximately 2 in 5 Canadians will be diagnosed with cancer in their lifetime, and approximately 1 in 2 of those will succumb to their disease, which makes it something we should really focus on and study. There are 2 types of cancer that I've been focusing on over the past 4 years. The first is breast cancer, which accounts for 25% of cancer diagnoses in women. And the specific subtype that I'm focusing on is triple-negative breast cancer, which has the worst survival outcomes, especially when it's metastasized, with a survival of less than 13%. The other tumor type that I've been working on is melanoma, which is an aggressive form of skin cancer. And again, once it's metastasized, the survival rate goes down drastically to less than 30%. So these 2 tumor types are really ones that need more advancement and to treat because they are so deadly. And in order to treat cancers, there's 5 main cancer treatment modalities that we use today. A lot of these have been around for a long time, and some are newly emerging. So starting off with surgery, it's the excision of tumors from the body. It's pretty basic, but there's a lot of morbidity and mortality associated with it. Radiotherapy uses radioactive materials to treat cancer. Again, a lot of morbidity. Cytotoxic chemotherapy is probably the most well-known therapeutic out there, which uses toxic agents, but unfortunately, these agents don't discriminate between healthy cells and cancerous cells, so there's a lot of off-target effects. To mitigate this, molecularly targeted therapies were developed, which target specific phenotypes of specific cancers in order to kill those cancer cells. And lastly, and the focus of my thesis, is cancer immunotherapy, which uses a patient's own immune system to fight off their cancer. So one main way of stimulating such an anti-cancer immune response is through a process known as immunogenic cell death. So to briefly walk you through it, what we have are agents that induce immunogenic cell death. So in this case, this is illustrating anthracyclines or cytotoxic chemotherapy and radiotherapy. Upon induction of ICD, you get a release of various tumor antigens and damage-associated molecular patterns. These then recruit and activate our innate immune system, specifically antigen-presenting cells, illustrated here as dendritic cells, which then go on to prime the adaptive immune system, illustrated here as CD8 and CD4 positive T cells. These T cells then proliferate and hone into the site of the tumor, where they can induce direct tumor cell lysis. My research has focused on the interaction between these antigen-presenting cells and the adaptive immune system, specifically CD8 positive T cells. And in order for there to be effective priming of these CD8 positive T cells, you need 3 main signals. The first signal is antigen presentation by antigen-presenting cells to the CD8 positive T cells. Second is co-stimulatory molecule binding between these 2 cell types. And lastly, in the focus of my research, is signal 3, which is the production of pro-inflammatory cytokines. With all 3 signals together, you get efficient priming of these T cells, which then become activated and are able to induce tumor cell lysis. So one way of stimulating this pro-inflammatory cytokine response is through receptors known as pattern recognition receptors. These were originally identified as binding to components of invading pathogens. So here, for instance, we have TLR4, which is a pattern recognition receptor that binds to lipopolysaccharide, which is part of bacterial cell walls. You also have other ones such as TLR7, 3, and cGAS-STING, which bind various components. And upon activation of this pathway, you get a signaling cascade that results in the activation and phosphorylation of IRF3 and NF-kappa B, which then translocate to the nucleus, where they function as transcription factors in the production of pro-inflammatory cytokines, namely IL-12 and interferon alpha beta. Our group and others have shown that FES may play an important role in regulating this pathway. Our group has shown that FES plays some role in endocytosing TLR4, thus preventing further activation of this pathway and shutting it off. And another group has shown that FES is able to phosphorylate and activate the tyrosine phosphatase SHP2, which functions at the level of TVK1 and TRAF6 to turn off this pathway. And so what we think will happen is if you knock out FES from an antigen-presenting cell, there should be an increase in the amount of pro-inflammatory cytokines produced, thus resulting in an increased T cell activation and hopefully better tumor control. So this leads me to the hypotheses of my thesis. The first is that FES deficiency will improve current cancer therapeutics and stimulate anti-cancer immune responses. The second is that FES deficiency will increase pattern recognition receptor signaling in antigen-presenting cells. Today, I'll be presenting the macrophage side of things, and promote pro-inflammatory cytokine production. And lastly, it'll improve macrophage-mediated T cell priming, cancer cell killing, and tumor control. So I have three main aims for elucidating these hypotheses. The first is to explore the impact of host FES deficiency. And I wanted to look how this would impact tumor growth, tumor immune profiles, and response to immunotherapies. The next is the downstream effects of FES. So we wanted to identify the immune cell types that actually have FES, the signaling cascades and the cytokines that are regulated by FES. And lastly is to look at the properties of FES-deficient macrophages. So we wanted to see the differential cytotoxic T lymphocyte or CD8-positive T cell priming, tumor control, and we wanted to look at this both in vitro and in vivo. I'll just be presenting, well, you'll see. So getting into the results, starting with our first aim, I want to bring you back to this diagram here where you have anthracyclines. In our case, we use doxorubicin, which induces immunogenic cell death, which we hope will activate the immune system and be able to cause delays in tumor growth and advantages in survival. So to explore this, I used a triple negative EO771 cell line and a B16F10 melanoma cell line, and grafted these cells into either wild type or FES knockout animals and treated them with either 5% DMSO or with doxorubicin. In the case of the triple negative model, we treated 3 times with 5 milligrams per kilogram. And in the melanoma model, we treated twice as often, but at half the dose. And so we measured tumors until an endpoint of 1.1 centimeters cubed or until they ulcerated. And so what we found is when you look at FES wild-type mice in black treated with DMSO and FES knockout mice treated with DMSO in green, you see that in both tumor models, there's a significant delay in tumor growth, as well as a significant improvement in survival in both these in FES-deficient animals. When you treat FES wild-type animals with doxorubicin in red, you see that in the triple-negative model, there's actually no tumor growth or survival advantages conferred by doxorubicin. However, in the B16 model, there is a slight advantage in both tumor growth and survival. What's interesting is when you compare FES wild-type animals treated with doxorubicin to their knockout counterparts that receive no treatment, you see that the tumor growth rates and the survival rates are near identical, suggesting that FES deficiency on its own is able to delay tumor growth to the same degree as doxorubicin in wild-type animals. So that's quite exciting. And then when you add in doxorubicin therapy into these FES-deficient animals, you see that there's an even greater delay in tumor growth and a significant improvement in survival, suggesting that doxorubicin is more effective in FES-deficient animals. So because doxorubicin is able to elicit an anti-cancer immune response, we wanted to see what was actually happening in these tumors. So to do this, we engrafted both cell lines into, again, wild type or knockout animals. We treated with an accumulative dose of 10 milligrams per kilogram per mouse. Then we isolated the tumors and spleens and looked at the immune profiles. And so when we looked at the T cell compartment of these tumors, while there's no difference in the number of infiltrating T lymphocytes, what we did see was a difference in activation. So in both models, when you have FES-deficient animals alone, there's an increase in T cell activation compared to their wild-type counterparts. And this is further increased when you add doxorubicin into FES knockout animals, but is not the case in wild-type animals. Furthermore, in both models, we see an increase in PD-1 positivity on these T cells. In the triple negative model, this requires a FES deficiency as well as doxorubicin therapy. However, in the B16 model, FES deficiency on its own is able to increase PD-1 positivity. When we look at the macrophage compartment, while again, there's no changes in number of infiltrating macrophages, we do see a reduction in both cases of M2 macrophages, which are pro-tumorigenic, and a subsequent increase in the amount of M1 macrophages, which are anti-tumorigenic. So these results all suggest that there's a shift from a pro-tumor microenvironment to an anti-tumor microenvironment in our FES-deficient animals. And something that really stood out, because it's an actionable target, is this increase in PD-1 positivity on the T cells. So just as a bit of background, PD-1 is an inhibitory receptor on T cells that, when bound to its ligand PD-L1, which is expressed by many different cancer cells, it suppresses the activity of the T cells and preventing its function. When you treat with an anti-PD-1 antibody, this sequesters away the PD-1 receptor, preventing the T cells inactivation and allowing it to go forth and kill the cancer cells. And what was exciting is 5 years ago, which was crazy to say, there was a study published in Nature Medicine showing that if you treat patients with doxorubicin, genes related to PD-1 and PD-L1 were upregulated, and those patients then responded better to anti-PD-1 therapy. So this is similar to what we see in our model, where doxorubicin increases the amount of PD-1 positive T cells in our triple negative model. So we thought, why not treat with anti-PD-1 therapy as well? So again, I engrafted both tumor models into either wild type and knockout animals. And this time, all mice received doxorubicin and then either received a control IgG antibody or a therapeutic anti-PD-1 antibody. And then we measured until endpoint. And what we found was that, again, when you compared FES, wild type animals treated with dox in black to knockout animals treated with dox in green, we see that there's a significant delay in tumor growth and a significant advantage in survival. When you add in anti-PD-1 therapeutic antibodies into the wild type animals, when looking at tumor growth, we see that there's a significant delay in tumor growth in both models. However, there's no impact on survival whatsoever. So this was kind of striking that this really potent therapeutic is not working in our wild type animals. When you compare wild type animals treated with dox and anti-PD-1 to FES knockout animals treated with only dox, we see that in the triple negative model, these knockout animals perform just as well as the dual therapy treated wild type animals in both terms of tumor growth and survival. But what's exciting is in the melanoma model, FES deficient animals only treated with doxorubicin actually perform better in both tumor growth and survival metrics compared to their wild type counterpart that were treated with both treatment modalities. And finally, when you add in anti-PD-1 therapeutic antibody into FES knockout animals, there's a significant delay in tumor growth and a significant improvement in survival, which is striking because in our B16 model, PD-1 therapeutic antibody had no effect on survival in the wild type animals, but now we're seeing a drastic increase in survival in our knockout animals. We were even able to cure one mouse in the triple negative model, which is kind of exciting. So moving on to the next aim, we're looking at the downstream effects of FES. I want to bring you back to this signaling cascade here, where upon activation of TLR4, you get this signaling cascade that results in signal 3 cytokine production, and that FES is able to inhibit this through different modalities. So we wanted to look at the levels of tank binding kinase phosphorylation, IRF3 phosphorylation, and NF-kappa B phosphorylation to see if there's a difference between wild type and knockout macrophages. So what we did is we isolated bone marrow-derived macrophages from either wild type or FES knockout animals, treated them with LPS for different times, and then looked at the signaling cascade via Western blot. And what we found was in our FES deficient macrophages, there's a significant increase in NF-kappa B phosphorylation, tank binding kinase phosphorylation, as well as IRF3 phosphorylation, suggesting that this pathway is more active in FES knockout macrophages compared to the wild type counterparts. Interestingly, our results also show that there's no difference in SHP2 phosphorylation upon LPS stimulation in this context, which is important to note, as another group showed that this was changed. We're still kind of looking into this because there's different contexts, or different contexts can have an impact on this, but in this model, at least, there's no change. So now that we've seen that there is a difference in the signaling cascade, we wanted to see how this impacted the amount of cytokines that were produced, and namely signal 3 cytokines produced by these transcription factors. And so we did the same experiment, except this time we isolated the supernatants and looked at cytokines by a Legendplex assay. And so I'm only illustrating 4 cytokines here, TNF alpha, IL-6, interferon beta, and IL-12. And what you can see is that at one time point or another, FES-deficient macrophages produce more cytokines than their wild-type counterparts, and in certain cases to a much greater degree. What's interesting to note, though, is that when we look at IL-12, which is one of the main signal 3 cytokines, we actually don't see a lot of cytokine being produced at the 12-hour time point. This is in the single digits of picograms per mil. So this made us think, well, what's going on here? And so we looked into the literature and found that IL-12 is a unique cytokine where it can be secreted, but it can also exist in a membrane-bound form. So while there might not be a lot of cytokine being secreted into the supernatant, there might be a lot of cytokines still associated with the cell. So to look at this, we again did the same assay, but we waited 24 hours. And then we looked at total levels of IL-12 as well as surface levels of IL-12. And when we looked at the total levels of IL-12, what we found was upon LPS stimulation and waiting 24 hours, there are more FES knockout bone marrow-derived macrophages that were producing IL-12 compared to their wild-type counterpart. And when you looked at only surface levels of IL-12, you see that upon LPS stimulation, there's an increase in the amount of FES knockout bone marrow-derived macrophages that have IL-12 on their surface compared to wild-type, which show no change. So this is suggesting that IL-12 is remaining bound to the cell, which might have impacts on activating T cells in some context. And just to show that this result was real, we also repeated it with TNF alpha. And so when we look at total TNF alpha levels following LPS stimulation, again, there's more FES-deficient macrophages producing TNF alpha, but we found absolutely no TNF alpha on the cell surface, showing that this effect is unique to IL-12. And so this brings us back to our signaling model, where FES might play a role in TLR, or we know that FES plays a role in TLR4 internalization. FES might play a role in SHP2 phosphorylation, but now there's a third role for how signal 3 is being presented to T cells, and it might play a role in the trafficking. And so by knocking out FES, we see that IL-12 might not be able to be secreted to the same extent, and will actually exist on the cell membrane and be able to be presented to T cells in that manner. Up here. So moving on to my third and final aim, we wanted to look at the properties of FES-deficient macrophages. And so to do that, I want to bring you back to this model here, where we have the interaction between the macrophage and the T cell, where we need 3 signals in order for there to be efficient priming of T cells. And so we wanted to look at this interaction here and see if FES-deficient macrophages were actually able to prime T cells to a greater extent than their wild-type counterparts. So to do this, we isolated CD8 positive T cells from OT1 animals. Now, these T cells are unique in that their T cell receptor, all the T cells have a T cell receptor specific for a peptide from chicken ovalbumin known as synfecal. And so we, at the same time, pulsed our wild type or knockout macrophages with the synfecal peptide and stimulated with LPS. We then co-cultured the T cells and the macrophages together, and then recovered the activated T cells and did flow cytometry looking for interferon gamma, which is a surrogate for T cell activation. So what we found is even in the unstimulated group of macrophages, FES-deficient macrophages were able to prime more T cells than their wild-type counterpart. And when you add in LPS to this co-culture model, you see that there's a significant increase in the amount of T cells that are activated. And again, FES-deficient macrophages were more efficient at priming these T cells compared to their wild-type counterpart. Not only are there more T cells being activated, the extent to which they're activated is also increased. following a co-culture with FES-deficient macrophages compared to wild type. And this is illustrated by median fluorescent intensity of interferon gamma in these T cells. So far I've shown that there's activation of T cells, and this exists in vitro as well as in vivo, but we want to see if in vivo the main driver is actually these macrophages. So what we did was we did a macrophage adoptive transfer where we engrafted either the melanoma or the triple negative model into our wild type animals. We then isolated wild type or FES knockout bone marrow derived macrophages, polarized them to an M1 state, which is anti-tumorigenic. And then we injected these directly into the tumor, allowed the tumors to grow for a bit longer, and then we isolated the tumors and took their weights. So we wanted to see if these macrophages could actually reduce tumor growth and if there's a difference between the two genotypes. And so what we found in our B16F10 model was that PBS treated tumors obviously had the largest weight upon resection. And when you treated these tumors or injected these tumors with wild type macrophages, we saw a significant decrease in the tumor volume when we resected them. But what was exciting is when you injected these tumors with FES-deficient macrophages, there was the greatest reduction in tumor volume. And this was consistent across both the melanoma and the triple negative model, where we also see a significant decrease in tumor volume compared to the wild type injected animals. So all in all, this brings us to a couple of conclusions that I've found during my PhD. The first is that host FES deficiency enhances doxorubicin therapy. Improved tumor control in the FES-deficient mice can be attributed to an increase in anti-tumor immunity. Host FES deficiency sensitizes both triple-negative breast cancer and melanoma tumors to anti-PD-1 therapy. FES deficiency enhances pattern recognition receptor signaling in macrophages, leading to increased cytokine production. And lastly, FES-deficient macrophages have enhanced T-cell priming and tumor control functionalities. So the impact and significance of this work is, first and foremost, FES is a novel immune checkpoint. When we take all this data together, it suggests that FES is actually an attractive target to inhibit that might enhance existing immunotherapies or exist as its own immunotherapy, which predominantly stems from the innate immune system. Next is that FES expression can actually serve as a predictive biomarker. Since we've been able to show that FES-deficient mice actually respond better to both doxorubicin and anti-PD-1, we might be able to translate this to humans and look at their FES expression levels to see which patients will or won't respond to anti-PD-1 therapy or doxorubicin therapy. A couple of future directions we have is we, again, want to translate this into patient populations. And so we want to perform or look for data sets that exist of single-cell RNA-seq that exist in triple-negative or melanoma cohorts of patients that have been treated with anti-PD-1 therapy to see if FES levels actually do correlate with response to therapy. The next is to engineer an oncolytic virus that expresses a microRNA against FES. And this has actually been done, and we're just waiting for the virus to arrive so we can start doing some experiments with that. So if anybody's interested, let us know. And then lastly, we're thinking about developing a small molecule inhibitor of FES. There currently exists an inhibitor, lorlatinib, that is able to inhibit FES. However, this is an off-target use, and there's a lot of other off-target indications of this drug, so it's not perfect. But we're hoping to develop our own FES inhibitor and bring that forward. So with that, I'd like to thank everybody who's contributed in my lab. Obviously, Peter, he's been okay for the past 4 years. My thesis advisory committee, my PhD defense committee, all my funding, and you guys for listening.
That's great. We've got half an hour for questions. I
Going
told
fast.
him that he could get the same talk as he gave, the 20-minute talk that he gave to his PhD defense, but just slow it down. Seems like that's too much.
Yeah.
So, questions?
Great job, Brian. Congratulations.
Thank you.
This was all very cool, and I'm not sure if I missed this at the beginning. Is there a reason that you chose breast cancer and melanoma specifically to use? And and like, could this be extended to other cancers, or is that something that you're working on, like looking at other cancers specifically?
Yeah, so our lab was, it's what we had. So our lab predominantly worked on breast cancer. And so that's where we started with this. But since this isn't specific to cancer cells, because cancer cells don't actually express FES, it can be translated to different types of cancers. So then at that point, we thought, let's move this into melanoma and see what happens there. And these two are actually quite interesting because immunotherapy isn't very as well studied in triple negative breast cancer. And it's not as successful as it is in melanoma. And so melanoma is a great model because there's a lot of success stories in melanoma, especially with immune checkpoint inhibitors, but there's still room for improvement. So that's one reason we went with melanoma. And then with triple negative, it's really not that successful. It's not used as first line in a lot of cases. So we thought, why not look at a really tough tumor to treat with immune checkpoint or other immunotherapies and see if we can improve both. So this can be translated to multiple different types of tumor, but you have to test each one individually because it depends on how these tumors arise. So we did some experiments in ovarian cancer, and we used the NF1 ID8 model that we have at Queen's. And so that's a hot tumor with a lot of immune infiltrates. And what we found was that that tumor model in our FES deficient mice actually did worse. And so in that case, it might be adding fuel to a fire. And if the immune system's promoting tumor growth and you knock out FES, it might make it worse. We stopped kind of exploring that after a while. But it can be translated to more tumor types. You just have to test each one individually. Yeah.
Charles?
Beautiful story, Brian. Congratulations. You delivered it beautifully.
Thank you.
Maybe I've asked you this question before, or maybe since I asked, maybe there's new information. The wild type mice, they express high levels of FES, I guess, and it seems to be constituted high.
Yeah.
So why do they do that? Why do they want to be immune suppressed? Like, how is, just to extend my question a bit, confuse everyone, how is, and I'm sure I've asked this question before, but maybe there's more information.
Yeah.
Now, how is FES regulated? Like, because you don't want your macrophages to be like checked.
Right. Yeah. Well, yeah, that's a good point. If you have FES always there, always suppressing that, then you can't get them to be activated and they're useless essentially. So we're not entirely sure how FES is regulated yet, but what I've been able to see, and I didn't present it here, was that when you treat these macrophages with LPS, RNA levels for FES actually go down. So upon being stimulated, the macrophage responds obviously and becomes activated. And one way it does this is by turning off FES to allow the cell to become activated and do what it needs to do. And this will probably come up later to suppress it, but that's one of the mechanisms. But how that happens, we're not entirely sure. But yeah, it's a good question.
So just to add to that, Charles, so these mice, remember, they're sensitive to LPS endotoxin challenge, but it's not like they all croak immediately if you give them any. There's obviously some regulation going on here, and it's not a binomial thing.
Yeah, because I imagine FES is important in fending infections as well. Downregulation of FES.
Yeah.
So you think it would have to be suppressed somehow?
Yes, it is somehow. But it's most likely a feedback loop of some sort that, yeah. I'm not too sure what's regulating it. But you need both high levels of FES at certain points, and you need low levels of FES at certain points. And so in a cancer context, there might be something that's driving it to have high FES levels so that it suppresses its activity.
So to properly target it, I think you we need to understand better. I mean, it's not for you to do this, but.
Yeah, I'm done. I'm out of here. Yeah.
It's gonna take another 4 years. To find out how fast it's regulated.
Madam,
Great job, Brian. Congratulations.
Thank you.
I was just wondering if you looked at all at like the impacts of your research on metastasis. Like you mentioned in the beginning, that's obviously where the problems with therapeutics come in, is when the cancer.
Yeah. So we did a pilot study, kind of one of the first things I did. And when you infest knockout animals, when you did a resectin weight model, there was no metastasis in the lung compared to wild type animals that did have lots of metastasis that were big. And again, the FES isn't expressed in cancer cells. So it's like a tumor niche kind of thing. So FES is also expressed in vasculature, immune cells, certain secretory cells. So it does have an impact on metastasis, but it didn't go further in depth on that. But something that could be done is immune profiling of the lungs to see what immune cells are infiltrating that are there, not there, activated or not. Yeah, so with that small experiment, we can say that there is an impact on metastasis, but I haven't really delved further into that.
Marco, go ahead.
Yeah, I'll echo the others, like really great job, Brian. I had a question about something you mentioned earlier about host efficiency in FES and immunotherapy response. I was wondering if you guys were able to look at, or if there are any germline variants in FES that tend to show up in cancer patients, and if that's something that you guys can maybe leverage to understand how FES efficiency is impacting people in real life.
Let's see if I can bring this up quick. I have the wrong presentation up, but so we did look at that, and we looked at the TCGA, and we looked for, let's see what this is. So the first thing we did is we looked at RNA levels from patients of FES and correlated this with survival. So patients with low FES do better in terms of overall survival and disease-free survival. But number one, this is at the RNA level. Number two, there's more than just immune cells in the tumor. So again, vasculature is in there that expresses, so this might skew it a little bit. So then what I did next is I went to TCGA. I looked for patients who had any kind of mutation in FES. So it could have even been a silent mutation, but any mutation in FES that was in healthy blood. So again, it's not associated with the tumor at all, and it's the immune cells. And what we found is in the yellow, those are the patients that have some sort of mutation, compared to the blue, which is patients who have no mutation in FES. And so again, this is across all cancer types. So it suggests that there are some mutations that arise in patients, and it actually confers a benefit. Now, what these patients are being treated with, I don't know.
Yeah, if I can have a follow-up, that's interesting because like TG obviously is, I guess, old now.
Yeah.
So like a lot of these people didn't get immunotherapy, so a lot of them had just like primary cancer or surgical resection.
Yeah.
That's really interesting.
And doxorubicin is used in breast cancer, is used in other cancers, so that could also be in there. We've tried looking for data sets that have single cell RNA-seq data of patients treated with immune checkpoint, but the detection level is like the depth isn't deep enough to actually pick up FES transcripts in any meaningful amount. So that's kind of something we're struggling with now to try and find that data.
Marco, I always look at your CHIP data, and the FES number appears as a gene in CHIP, but then I don't know if it's covered. But most of that is got whole genome, whole exome sequencing now, right?
Not targeted is better, but whole genome and whole exome still comes up in like the bigger population things, and TCG and stuff.
Yeah.
And just to go back to your question, this is the metastasis data. When you do the resectin weight, you see that there's no or very little metastasis in the knockouts compared to the wild type. Yeah.
Any other questions? Oh, she-
This is kind of not necessarily cancer, but so have you seen if that's levels vary across autoimmune conditions?
So I haven't specifically looked into that, but again, it would probably have an impact. So I think there's a couple papers potentially, but so as we've been saying, FES actually controls the immune system, and if you don't have FES, you get a lot of cytokine being produced, a lot of immune activation, which is a hallmark of autoimmune diseases and also things like sepsis. So the thought is it probably plays a role because if it's not there, your immune system will go unchecked in certain aspects and can start targeting itself. So yeah, it's not something I've looked at, but it's definitely possible. I think people have theorized that it would play a role in that.
That would be a good one to parse the UK cohort on the basis of, because, you know, we look at
Yeah.
that whole big 450 or however many thousand, we'll call them patient subjects in that cohort. And you don't show that, but
No.
there are a couple of SNPs in there that we've been looking at, but that's looking at what, probably 50,000 cancer patients out of that whole group.
Yeah.
But, it'd be interesting to look at something like autoimmune.
Yeah.
Anybody else got a question? This is last chance to get this guy. He's going to some place on Baffin Island or something. All right, well, if not, thanks very much.